My name is Ned Willig, and I'm with the Great Lakes Commission. It is my pleasure today to have presenting here Sylvia Jacobson from the University of Maryland. Before we jump into the presentation, I just want to share a little bit about the Great Lakes Phragmites Collaborative and who we are, and then we'll hand it off to Sylvia. So the Great Lakes Phragmites Collaborative is a joint effort between the Great Lakes Commission and the U.S. Geologic Survey, and the collaborative is a partnership designed to link people, information, and action on addressing invasive Phragmites in the Great Lakes region. What I'm going to do today is hand this off right now because it seems like I'm having some bandwidth issues. So Sylvia, if you can go ahead and share your slides, we will jump right into the presentation.
Yes.
If we can just quickly, Sylvia, if you can hear me still, I just want to do a quick introduction for those that can hear me, and then I'll let you go for it. So okay. I'm sorry, not as smooth as we'd like. I apologize for the bandwidth issues that we're having right now, but it's our pleasure to welcome Sylvia Jacobson today. Sylvia is a master's student at the University of Maryland, College Park, in the Environmental Science and Technology Department. She works with her advisor, Dr. Andrew Baldwin, as well as researchers from the Smithsonian Environmental Research Center, Dr. Dennis Wiggum and Dr. Melissa McCormick, as well as Dr. Corinne Kettering from the Utah State University to investigate native plant restoration in tidal wetlands dominated by the Phragmites australis. She graduated with a Bachelor's of Science and Engineering in Environmental Engineering from Princeton and worked on the remediation of contaminated waterways for 2 years as an environmental scientist with Anchor QEA. So thank you so much, Sylvia, and we're really excited to hear your talk, and please take it away.
Okay, thank you so much, Ned, for the introduction. I'm sharing my slides now. Can you see okay?
Yep.
And hear okay? Okay, great. So yes, as Ned mentioned, I'll be presenting on my master's research project on what happens after Phragmites is killed, specifically how native plantings can affect tidal wetland recovery following Phragmites australis removal. And this is an ongoing research project. We've now completed year 1 of the 2-year project. And I want to thank everyone who's been a part of and has supported this project. Ned mentioned some of them, the PIs on the project, my advisor, Dr. Andy Baldwin, Dr. Dennis Wiggum, and Melissa McCormick from the Smithsonian Environmental Research Center, and Dr. Corinne Kettering, who introduced me to you all at the Great Lakes Phragmites Collaborative from Utah State University, as well as Eric Buell with University of Maryland Extension, and also the many students and volunteers who have helped in this project, including from the Severn River Association and from Maryland Sea Grant, who has funded this research project, as well as my graduate research fellowship. So thank you. So I'm guessing that many of you all on this Students of Phrag webinar are familiar with the 10-foot tall grass species, the invasive Eurasian lineage of Phragmites australis, which has expanded widely in wetlands across the United States. Our research project is in tidal wetlands in Maryland's Chesapeake Bay region, where Phragmites has been prevalent and really exploded since the 1970s. In a recent EPA National Wetland Condition Assessment, they found that Phragmites was the most commonly observed wetland plant species in Maryland. And this rapid expansion of Phragmites australis into wetland ecosystems can strongly affect wetland ecosystem functions, including displacing native plants as well as certain estuarine animals that depend on those plants. Although I think it's always important to mention that there's also research showing that Phragmites can provide some beneficial ecosystem functions. Some animals can live in Phragmites, and notably in these tidal wetland areas, some research suggests that Phragmites may promote resilience to sea level rise and help store carbon because it produces so much above and below ground biomass, this may help elevate the elevation of the marsh. So out of concern for the native wetland plants and animals that Phragmites can displace, land managers all over the country and also in the Chesapeake region have gone to great lengths to try and get rid of it. One study estimated that over $4 million a year was spent on Phragmites removal in the United States. But the eradication of Phragmites doesn't always have the desired effect of native wetland species returning. Sometimes it does, sometimes it seems to work, but other times Phragmites can return within a few years. And one thing we've noticed in some of these tidal wetlands in the Chesapeake Bay region is sometimes you even can have the marsh collapse and no vegetation can return. So the goal of this project is to assess the role of planting native wetland species after removing Phragmites to see how that might help stabilize the marsh and accelerate the return of other native wetland species. And we're also interested in the different factors that can influence the different restoration outcomes at different sites, how the hydrology and soil properties, as well as the specific Phragmites removal intervention and the timing of planting native wetland plants after removing Phrag and how that affects these different outcomes. So we have two sets of research questions. The first set is on how planting native wetland species can affect the recovery of tidal wetlands after removing Phrag. So specifically, which native plant species can grow after Phragmites is removed, and what are the different functions of those native species? How do they store carbon? How do they promote soil strength or support soil oxygenation? And we also were interested in how planting design affects plant growth in order to help make recommendations to land managers. So you can see in the picture on the right, we had planted native wetland species in both more clumped and dispersed planting arrangements, wondering if the more close, denser plantings may facilitate each other's growth and help oxygenate each other's root zones in these stressful flooded wetland conditions, or maybe they would compete with each other if grown too close together and inhibit each other's growth. The second set of questions is trying to understand why the restoration outcomes are so variable after Phragmites is removed. And this is in part motivated by what we're seeing after year one of this 2-year experiment, where we planted native wetland species at different sites after removing Phrag. And at some sites, like in the picture at the top, the native plants became established and grew well, where at other sites, like this one in the picture at the bottom, not only did the native plants we planted not grow, but no other vegetation grew back either. So we want to understand the different environmental factors that affect these different results, including the hydrology, salinity, and soil biochemistry at different sites, as well as how the timing of our planting affects native plant recovery. So in the site at the picture at the bottom, Phragmites was removed over 2 years ago. And so we think that the rhizomes may have decomposed, leading to highly reducing stressful conditions for plants, where the other sites, like in the one at the top, we planted native species right after removing Phrag, so perhaps that timing was more conducive to plant growth. And another question we're asking this coming season is we're trying something new where we're adding sand to the soil for some of our plantings before planting native wetland species, since that's a common practice among wetland restoration practitioners, and they seem to find it has beneficial results. So we want to understand, we're going to do that to half our plantings and see how that affects their growth in these very mucky soil substrate sites, and also try and understand by what mechanisms that might affect plant growth. So to answer some of these questions, we have a field experiment at 12 study sites in tidal wetlands along 3 different Chesapeake Bay tributaries. You can see this is the map of Maryland with the Chesapeake Bay running through the middle, and we have 3 sites along the Severn River, 3 sites along the Rhode River, and 6 sites along Parker's Creek at the American Chestnut Land Trust in north southern Maryland. And this is the layout of our experimental design at each site. We cut back Phragmites and have 4 experimental plots, and 3 of which we planted a different native wetland plant species, and 1 of which is a control plot where we killed Phragmites and didn't plant anything. And at some sites, we also have a Phragmites reference area where we have an undisturbed stand of Phragmites for comparison of ecosystem functions. Although we only have this at a few of our 12 sites, as at some sites, community groups and others in the project wanted to completely remove Phrag from their wetland area. And here's a picture of our planting design, how we planted both clumped and dispersed plantings. The clumped ones were spaced 10 centimeters apart, dispersed ones 33 centimeters apart, about a foot apart. And so here are the species we planted at our low salinity oligahalian sites. We chose all clonal perennial plants that we thought could grow quickly, become established, and produce a lot of below ground and above ground biomass to help stabilize these sites after Phragmites is removed. So one of these species, the Lineage oryzites, rice cutgrass with the X through it, we planted at the beginning of our experiment, but it all died within a week or 2, so we replaced it with a different species, Spartina sinusoides, big cordgrass, and we're not sure. We think that it may be because the Lineage oryzites we got from the nursery was not looking good at the time we planted it, so it's hard to say whether that was because of our specific plantings or something about these species. But these are also all species that grow near our study sites. We see them in the surrounding wetlands. So we also had Pelchandra virginica, arrow arum, and Panicum virgatum, switchgrass. At our more medium salinity brackish sites, we also planted clonal wetland species, but ones that were more tolerant of more brackish conditions, including, we also did Spartina sinusoides, big cordgrass, and we planted Displace spicata, saltgrass, as well as Spartina patens, marsh hay cordgrass. So this June, actually, next week, we're planting more species, including some of the same ones as last year, to see how they might grow differently in a different year, as well as testing some new species. So we're again planting Spartina sinusoides, big cordgrass, Displace spicata, saltgrass, and Pelchandra virginica, and we're also testing Spartina alterniflora at our brackish sites because we know it's very tolerant of higher salinities and of high levels of flooding, so it might be able to survive at some of our very flooded sites where nothing seems to be growing. And we're also planting Hibiscus moschutos at the fresh, low salinity sites, which is a perennial plant that grows in the area, known to be very hardy, so we want to test out how that might do. So I'll show some pictures of what our study sites look like. These are study sites along the Severn River. You can see we cut back Phragmites and we lay out black plastic to prevent the Phrag from growing back. And then out of the plastic, we cut out areas for our study plots where we planted native species. This picture in the bottom left is a drone image of us planting native species at the site. We have 3 medium salinity sites along the Rhode River at the Smithsonian Environmental Research Center. Here's a picture of us planting the native wetland species there. And we also have 6 sites at Parker's Creek at the American Chestnut Land Trust, 3 low salinity, 3 medium salinity. This picture on the right, a fellow grad student took with a drone, which shows the aerial view of the creek, a tributary of the Chesapeake Bay, from the headwaters to the mouth, and the sites are along a salinity gradient as you head towards the Chesapeake Bay. We also have a mesocosm experiment, which we call Marsh Organs, where we have potted plants at varying water levels. And we planted the same species we planted last season in our field study, as well as Phragmites in the pots. And this helps us isolate the effect of inundation on the plant's functioning, growth, and ability to store carbon, among other things. And we have one lower salinity marsh organ and one medium higher salinity marsh organ. So some of the measurements we're taking to try to understand which plants can grow and why and what affects their growth. This past season, we looked at plant cover, plant growth. We measure water levels at all our sites. We have monitoring wells at each site, as shown in this picture at the top, where we have a data logger that records the water level throughout the growing season. We measured soil oxidation reduction potential and pH to see how reducing the different sites were. And then at the end of this coming growing season, we'll be harvesting all our plants, measuring above and below ground biomass to see how much biomass they produced, how well they became established. We'll also be assessing the soil shear strength of the different sites using a device as shown in the picture at the bottom left, where you have 4 prongs that you insert into the soil, and then you spin the device with a torque wrench, and the amount of torque takes to spin is related to the strength of the soil and the roots. We also were interested in measuring sedimentation in our different sites. Undergraduate researcher is shown in this picture at the bottom right, putting out sediment tiles, which we used to measure how much sediment accrues at different sites, and we put them in areas with Phragmites and areas without Phragmites to see how that affected sediment trapping. We're also interested in the soil biochemistry of our different sites and how that may be affecting plant growth and restoration outcomes. So we're considering looking at the soil microbial communities to compare functional groups to see if we may have iron reducing or sulfate reducing bacteria, as well as measuring some of the chemistry more directly. For example, testing if there's sulfide in the soil pore water at some of our brackish sites, which we think might be part of the reason also we're not seeing a lot of plant growth at some of those more brackish sites. So some preliminary results from year 1 of our 2-year field season. As I've mentioned, we had a lot of variability in native plant growth, and a lot of sites, native plantings did not grow well. These bar charts show the percent cover at the end of last year's growing season across our different sites for each species. And you can see for our low salinity sites, only at one site did the plants really grow well and become established. And then at our intermediate salinity sites, at all the Rhode River sites, the plants grew well and became established. But the plants did not grow well, and at these 2 sites, they did not grow at all at Parker's Creek. And we did not see a notable difference between our clumped and dispersed planting in the first year. It seemed that either both clumped and dispersed plantings grew well at a site, or neither did. Just here's a picture of what this variability in plant survival and growth looks like at our different sites. This is from this past May, how the sites are doing. So here's one Parker's Creek intermediate salinity site where no native planting survived and no native volunteer plants are coming in to recolonize the site after Phrag was removed. And here's a site along the Rhode River where we have Disticulus spicata planted on the left and Spartina patens on the right. And these plantings are doing well and continuing to grow now in the 2nd growing season of the experiment. So we think that some of this variability is due to the hydrology at the different sites, where we've noticed the driest sites had the highest plant growth. This figure shows on the x-axis the proportion of time that a site was flooded above 15 centimeters, and then the percent cover of our planted species averaged across the different species we planted. And the plants grew well at these Rhode River sites, which were a lot less flooded, as well as this one Severn River site that was less flooded. But we don't think that that explains all the variability in native plant growth. We think that soil substrate and biochemistry may also be playing a role, and this is something we're trying to investigate this summer and this field season. So for example, here's a picture on the left of one of our Parker's Creek sites, where you have this very soft, soupy, mucky substrate. And this is a site where Phragmites was removed about over 2 years ago. And we think that the Phragmites rhizomes may have decomposed in this time, and increasing this, leading to this very reducing low oxygen environment that might be stressful to the plant growth. And then in this picture on the right, here's one of our Rhode River sites and how it looked right before we began planting. And the substrate has more structure following the recent cutting of Phragmites. So we think that maybe that structure and perhaps the oxygen in the soil from the presence of the Phrag rhizomes and more oxygen, since they haven't yet decomposed and microbes haven't yet consumed as much of the oxygen, might have been more conducive to plant growth. And one thing, additional thing we're testing related to enhancing plant growth in these mucky soil substrates is how adding sand to the soil may affect the native plant growth. We've heard from a lot of colleagues in the wetland restoration industry that they commonly add sand to wetland restorations, that they find that that can be very effective in increasing plant growth. And we're not completely sure of the mechanisms of this, whether that's increasing soil drainage with the higher porosity, or it's less organic material in the sand relative to these other or highly organic soils. So you may have less reducing conditions, perhaps something, the structure of the sand. We're not sure how that would affect things, but we're going to, this season, we're going to plant half of our plantings with sand and half without, and test the effect on native plant growth, and also take different measurements to try and understand how the sand affects the soil environment. So I was hoping that if anyone has any thoughts, it's still an ongoing experiment, so I'm open to ideas and suggestions also, or any questions about this research.
Awesome. So yes, we if you have questions, feel free to type them in the chat box. Or if you also want, you're also welcome to raise your hand and I will go ahead and unmute you. Our first question is coming from Holly Best, and she was asking, the Phragmites was just cut, not sprayed with herbicide. Is that correct, Sylvia?
Yes, we just cut it back and we put out black plastic, which people call solarizing Phrag, but we did not use herbicide at our plots.
And how did you choose the plots relative to kind of the elevation in the marsh? Are these all kind of similar area relative to where the water, like the tidal fluctuations, or is there variability there too?
So there is variability. So we chose sites that had small patches of Phrag that in these tidal wetlands that different community groups were interested in participating in this study. So they're all pretty close to sea level, and in similar ranges relative to the tide, but there are differences in flooding where some of our sites are more flooded than others, and that is one variable we're interested in seeing how that's affecting the plant growth. And that's part of the reason for the marsh organ experiment, is try to better understand the effect of the flooding.
Gotcha. And I noticed you talked about a couple of different species. Can you talk a little bit more about how you selected those, or if you'd given consideration to other ones that might have different rates of different growth rates or different characteristics?
Yeah, so we selected species. One, we wanted all clonal species, perennial species, things that could grow rapidly, and also species that were already at these sites that we see in the surrounding wetlands that we think would then likely do well at those wetland sites.
Great.
We did consider other species I'm trying to think. We considered a few other species, but we thought these ones were at these sites. They were clonal and also most accessible for growing at the greenhouse in Maryland. So a combination of factors.
Gotcha. And with the planting, in some of the previous talks we've had have talked about like the density of planting and how close together you clumps. And you talked about having clumped versus dispersed, but with the clumped, was there any thought of did you notice any impact on if stuff was clumped closer together versus farther apart? Maybe that's something you didn't measure, or just any consideration on kind of the density of species as you planted them.
Yes, so that was one thing we were measuring was with the planting design and the clump dispersed. So this year we assessed plant growth and cover, and we didn't see a noticeable difference between the clumped and dispersed in year 1 of the experiment, although we'll continue to see in year 2. We did measure soil redox potential also in the clumped and dispersed areas, thinking that maybe the more clumped plantings would oxygenate the soil, have less reducing conditions, but we didn't see a difference in year 1 related. It was just the redox potential was very reduced at all our sites, but very variable. So we did not see any trend.
We have any other questions for Sylvia? Any ideas from folks that are either in the Great Lakes region or in the Mid-Atlantic? I know we have a variety of people on with different perspectives, so we'd be happy to hear more thoughts. In your sites, Sylvia, did you see any reinvasion of the Phragmites after they had gone, after you had removed them in the year, or was?
So, yeah. A little bit. So part of our experimental method is that at all our sites we're continuing to manage the Phrag and cut it back throughout the experiment, although mostly we don't have to do that. It actually seems to have mostly stayed out of our plots. I mean, at some sites there's a difference where some sites we've completely eliminated the Phrag because it was a smaller patch, and where the circ sites, the ones at Smithsonian on the Rhode River, for example, where we cleared Phrag, but it's still surrounded by Phrag, so those ones have a little bit more Phrag returning. But we still wanted to test the native plant method at those sites.
Great. Yeah. Are there any plans to look at similar results with the native plants following a herbicide application?
Well, one additional part of the project, which I didn't focus on in this talk, is we're also doing a retrospective analysis of other areas in the Chesapeake region where stands of Phrag have been treated, and some of those have been treated with herbicide, and trying to look at long-term results and compile a database of how different sites are recovering many years later after different kinds of Phrag managements.
Interesting. So thinking, how do you expect these results to kind of inform the work thinking about restoration in the face of climate change? How would these results apply to managers with those considerations?
Well, I think especially some of these tidal wetland sites are becoming increasingly more flooded, which is maybe part of the reason why when you remove Phrag, it's hard for native species to become reestablished at these already stressful sites that are becoming increasingly more stressful and flooded. So I think understanding what the outcomes of Phrag removal will be in the face of sea level rise, even if it's not the outcome you want, is still important. And maybe, we're seeing if native plantings could have a role, or maybe elevating the marsh with sand might help in some of those areas that are very flooded. But at least to know, to have a sense of what would happen, to be better informed.
Yeah, so that actually is a great, it reminds me of another question I had on the study design. You had, it looked like the control was with the Phrag removing, and it sounded like you had some study of the Phrag stands that were adjacent, that had not been removed. Did you track any, were those a factor in the consideration just to see the differences between biogeochemistry or water movement in those Phrag stands versus the ones that had been cleared?
Yeah, well, I think that is part of the story when trying to understand Phrag management, is to know, I mean, we're looking at if you plant native species after it's removed, and if you don't plant native species, but also to understand characteristics of the undisturbed Phrag stand. A lot of other studies have focused on that, so that's why it wasn't as much of a focus for our study, but still having a few sites where we can measure, look at the soil redox potential and look at carbon storage in those Phrag stands, and then comparing in areas where we've removed Phrag and areas where we've removed Phrag and planted things, to compare how the ecosystem functions in the marsh in those different areas, and try to understand the changes that are happening to the marsh. I mean, ours is a short-term study, so in 2 years you may not recover all the ecosystem functioning of a Phrag patch just by planting native species, but which is kind of why we're more interested than in just comparing the native species to where Phrag was removed, to see if it's promoting the desired ecosystem functions and the soil strength and the carbon storage in a positive trajectory.
Well, that's really great, and I think this is really fascinating research. I will give one more chance for people that have questions. Feel free to either type them in or raise your hand, and you can come off mute and ask them yourselves, but we'll give one more moment for that. We will be this webinar has been recorded, and we will have a recording following, and that will go out too. We will, Sylvia, if you're all right, I will also share your email. So if anyone has any questions on the design or any of your results or research going on in your region, they would be able to contact you that way.
Yes, do feel free to reach out.
But otherwise, we really appreciate everyone joining today. And again, thank you, Sylvia, so much for presenting and hanging with us through some technical difficulties. But
thanks so much for hosting me today. It's a pleasure to join you all.
Yeah, yeah. So best of luck with all of your research, and thank you everyone for joining, and we will be in touch soon with a webinar recording. So thank you all and have a great day.
Thank you.